Okay, I see the
And
slides. I just heard you, so it looks like we're good. Next, we've got a talk from Andrea Stocco and Catherine Seibert from the University of Washington. Go ahead, Andrea.
thank you. We're going to try to actually team tag in this talk. And we tested it, so we should be without problems, but please bear with us if we have any kind of accidents. We're kind of like the closing talk in this series of neural talks, and it's difficult to follow up to crystallize with the entire theory, but we're going to try to do our best. And we're going to take a very different approach. So we're going to go from like the very micro architecture of the brain to the very macro architecture of the brain, all the way up. And first, I want to dispel some common misconceptions that are still common in the world of people who are a little bit familiar, but they don't do neuroimaging as a habit or as part of their work. If you think about neuroimaging research, you might be thinking of something like this. People get into the lab, they get their head put into an MRI machine, and then people do this type of brain mapping experiments where they compare, for instance, like a task conditions versus another conditions, and they find tiny specks of the brain that are more active in one condition versus the other. Now, that is still very true, but the truth is that this is kind of like old-time, old-fashioned neuroimaging. And nowadays, people have a slightly different way of doing neuroimaging that is a little bit more sophisticated and more relevant, actually, to the topic of this conference. And it looks like this. The first part is the same, of course. People come to the lab, go to a hospital, and actually get their head into a scanner. But then afterwards, instead of just doing contrast, people just look at the time series of these different parts of the brain, and they start analyzing how these time series evolve over time, and what is the correlation between these time series at different parts of the brain. And this is interesting because it let us observe like what is the time course of this activity, what are the networks that spontaneously form between different parts of the brain. And this is also something that varies depending on task demands, age of the participant, and other interesting aspects. Now, this type of analysis is called functional connectivity. And to the best of my knowledge, is the thing that neuroimaging people do that is the most relevant to cognitive architectures. And there are many reasons why this is relevant, but I think that this is the reason that I think will bring home the message for most of you. So if you take the brain and you divide it into 10 different aspects of activity, you can basically analyze the time series and create a correlation matrix. Now, the top part of this figure, the thing that looks like a tablecloth essentially, is this connection matrix, and this is what is called the connectome. Now, if you look at the connectome, this is pretty stable over time. All the brain regions, independent of what you're doing over time, show this spontaneous coordinating activity over time, even when you're sleeping, even when you're doing nothing at all. They also divide the brain into a set of functional networks. If you look at this tablecloth again, you're going to see that there are big squares and areas that are actually empty. And this is because the correlation between certain parts of the brain, the parts of the brain that in the brain figure are actually colored with the same color scheme, these parts of the brain tend to go up and down in tandem together all the time, or actually tend to be actually anti-correlated for different things. This is because they are functionally acting together or not. And they are also, in many ways, these networks form the fingerprint of an individual. They are very stable. You can actually use to train a classifier to recognize different individuals, and they're very sensitive. You can use this to make predictions about what a person will be like 10 years from now, for instance, where there is early symptoms of disease or the onset, for instance, of Alzheimer's disease. So if I were to summarize this in a single sentence, I would say this part here that I circled is the closest thing the neuroimaging can give us to the architecture of a person's brain. And this is, of course, relevant to us, right? Now, a few years back, we didn't have enough data available to actually investigate the problem of the cognitive architecture using this technique, because this requires a lot of data. But in the past 10 years, people have started pulling together data into huge databases, which are technically called brain observatories. And they've grown over time. And I just made a very short list of probably the most relevant projects. We have the Human Connectome project. We collect incredibly high quality data from 1,200 participants. And this is actually going to be the focus of Catherine's portion of the talk. We have the ABCD project. If you're interested in cognitive development, it collects data from 10,000 adolescents. We have the Open Neuron project, which is a free data set from which people can contribute their own data sets if they want. Our lab has contributed some. And if you really want to go big, The UK has recently released the Biobank, where they actually released data from 500,000 British citizens, including, of course, their anonymous data sets, but include like high quality structural, anatomical, and functional MRI data. So now the data is available. We have this exciting opportunity to use this data to test things that a few years ago were just like unthinkable, like what is the best way to describe the brain architecture? Can we learn something from brain architectures or not? And in this talk, what we're going to do is just to tell you two different stories, but I think there are two different stories that summarize fairly well the interesting relationship that exists between cognitive architectures on the one hand and these large repositories of brain data on the other. And I think these relationships are getting and growing to be very symbiotic. On the one hand, we have the possibility of using these large repositories of brain data to ask questions that were not possible to ask a few years back, like what is the best way to describe brain architecture? On the other hand, there's a way to take what we have learned about cognitive architectures in developing and refining cognitive architectures and bring it back to the world of neuroscience and use them as a new tool in making exciting discoveries in brain science and clinical neuroscience. I'm going to start with the first part of this presentation.
All right, so I guess the first question that we really wanted to ask was, what can we learn about large-scale neuroscience data from cognitive architectures? And I probably don't need to tell anybody in this audience that in terms of cognitive architectures, there's a lot to choose from if we wanted to try and mess around with it. But there isn't a whole lot of connection with neurological data in many of these models, and when it is incorporated, it tends to be sort of from a bottom-up perspective. And we wanted to see if we could take the opposite approach. Could we examine this neurological data using this high-level structure provided by an architecture? And so what we decided to look at was the common model of cognition, which is another thing that I probably don't have to describe a whole lot to this audience, but in case you've just decided to tune in for the final talk in this session, the common model of cognition tries to be a consensus model that captures a lot of the theoretical components and connections reflected by some of the more functional and architectures that can be used to create actual models and simulation and produce data. We really like this sort of high-level model for a couple of reasons. First, because it's very general, so we're using it to look at human brains, but in theory, because it's not tied to anything in particular, the same predictions should apply to AI, robots, or other intelligent agents. And it's also very minimal. We can't go create a model and run it in the CMC, but instead we can use the high-level structure as a means of guiding a top-down search for brain architecture that we haven't really been able to find from the more bottom-up approaches of network connections between brain regions. So the first step of this process is to map the components of the common model onto corresponding areas of the brain. And these mappings are pretty straightforward. If our model is predicting that there should be activity in the perceptual component, then we should also see activity in the perceptual areas of the brain. And from here, that tells us where to look in the brain. And now we want some actual brains to look at. So we grabbed a whole lot of data from the Human Connectome project, but we didn't actually need all of it. That was a little too much to chew for the first test that we did. So we took about 200 of the participants. We looked at just their task data and just their fMRI data. And we used only 6 of the 7 tasks. But even if we ignore the motor task, the other 6 tasks are still covering quite a wide range of sort of cognitive skills and components. And if we can have a model that is providing a good general account of the structure of the brain, then it should provide good predictions across sort of this full spectrum of tasks. We think that the common model will do a good job of this, but we would like to formally test it. So the way we're going to do that is we are going to try and compare the predictions of the common model on this task data and these individuals against the predictions of alternative architectures. And if the common model is the sort of better structure, then we should be able, it should provide a better account of the brain activity across the tasks and across all of the subjects. So for our alternative architectures, we went back and looked at the exhaustive list of cognitive architectures already existent and found that in general, they tended to fall into rough groups and categories. The largest of two of which were a hub and spoke architecture, where there is one central component that sort of drives activity among all of the other components, but there are no intermediate connections between sort of the spoke regions. Or architectures could be in a more hierarchical format, where activity sort of moves through all of the components, beginning with perception and ending with action. So from these two families, we came up with 3 plausible alternative architectures that use the same 5 sort of brain region / components used in the common model, but are connected using either the hub and spoke pattern or the hierarchical pattern. So the common model is shown in the red brain, and the other 6 architectures are shown and how similar they are to the common model. So a solid black arrow is a connection that is present in both the common model and the candidate architectures. A red arrow is a connection that is unique to the candidate architecture, and a dotted arrow is a connection that is absent in the candidate model but present in the common model. So we can see that a few of these models are very similar in structure to our common model, and some are very, very different. So with these structures, we now want to see if we can predict the brain activity in the tasks from our Connectome data. And if we were doing a traditional fMRI analysis, we would use a general linear model where we would identify some areas of the brain where we think there's going to be activity, say the blue circles in the brain, as well as some external factors that may have an impact on that brain activity, like the task conditions. And what we would be fitting is a set of parameters that reflects how much an external factor is affecting activity in the relevant region. However, we have 7 different architectures at this point, but they all have the same regions of activity and the same external factors, which means that if we use this method, we're going to end up with the same parameters for all 7 of our models, and that's not really good if we want to compare them. So instead, we're going to use dynamic causal modeling, which in addition to identifying activity regions and external factors, also takes into account the connections between those regions and how activity in one region may affect activity in another. And we get a much bigger set of parameters that are reflective of all of these connections, which means that when we fit the data with each of our 7 models, we'll get a different set of parameters that we can then compare to each other. There are many methods through which we could make this comparison. We went with a Bayesian approach that returns the probability that any given model is true given the set of data within whatever set of models that we're testing. So for each model, we get a distribution that looks something like this, where the location of the distribution on the x-axis essentially reflects how predictive the model is, how likely it is that that model is going to best predict a random subject, and that expected probability is sort of the peak of that distribution. Which all helps us make sense of this figure, which has many, many things in it, but shows all of the distributions for each of the 6 tasks for each of the candidate models. And there's a lot going on in here, but the main punchline is that we can see there's this red distribution that is consistently the furthest along the x-axis, indicating the highest probability that this model is providing the best account of the subject's data. And often it is quite a large margin away from any of the other candidates. And very gratifyingly for us, this red distribution is the common model and the predictions of it. So I guess in the interest of time and getting to the third part of the talk, I will just pause here to say that so far, at least of what we've tested so far, the common model provides the best account of brain activity across multiple subjects and multiple tasks, at least of the structures we've tested so far. But we have two main roads forward that we would like to take with this finding. The first is improving and expanding on the model analysis by incorporating resting state data, some modulatory connections between regions as opposed to just direct connections, and so on. And we would also like to see if there are practical applications for these predictions and models beyond just predicting activity in healthy adults. And I will hand it back over to Andrea to talk about exactly those things.
Sorry, Andrea, you're still muted.
Sorry, my bad. I didn't realize that. I thought I had it on all this time. Let me restart. If you are like me, a TV nerd, you're probably familiar with the character King Ezekiel from The Walking Dead. And you probably remember that its motto was drink from the well, replenish the well. And I take it to mean that if you are working at the cross between two different fields, like we found ourselves doing most of the time, you want to take some ideas from one field, but you also want to bring back some ideas. In this case, we want to use brain data to test our ideas about cognitive architectures and, you know, see if the space of the common model is actually a good place to work with. But we also want to use the common model to see if we can bring something back to the field of neuroimaging analysis. And this is going to bring me back to an example that I want to illustrate right now. So before getting into the details, I'm going to give you right now the main topic, and it's Parkinson's disease. And before explaining why Parkinson's disease, I just want to summarize. Many of you are familiar with this, but I just want to tell you this is a disease that comes from the death of neurons in the midbrain that produce dopamine. So what you see here is the progression of the disease going from like a healthy brain to on the left to an unhealthy Parkinson's disease brain on the right, going to an early stage of the disease in the middle. And the red part that disappears slowly is the dopamine producing neurons that slowly die out as a progression of the disease. The result of this is, of course, like the motor symptoms that we see in Parkinson's disease and the cognitive symptoms that happen later on because of this. Now, why am I interested in Parkinson's disease? Because it provides a clinical neuroscience application. And we have been talking a lot in the symposium about health and mental health and physical health application. In many ways, clinical application has a strong translational test of, you know, any kind of like application or computational thing, because this is a critical mission application. Also, if you want to bring something back to neuroscience, generally speaking, clinical neuroscientists are probably the people in the field of neuroscience that are most open to collaboration with the field of computer science, machine learning, and AI researchers. If you were to rank them in terms of like openness to collaboration, I would probably put the clinical neuroscientists at the very top. They're excited about anything that can bring back something to their field in terms of like, okay, is this helpful to diagnose patients? Is this helpful to discriminate between patients and healthy controls, or tell something about the progression of a disease? But there's also something about Parkinson that is actually interesting in itself and provides a test for us. It is a very known and well understood etiology. So we know where it starts. It starts in this part of the brain that I showed you right in the previous slide, and that we have mapped in the common model of cognition. So we know, roughly speaking, which component is affected in Parkinson's disease that is mapped to a functional component in the common model of cognition, the procedural memory. It has very apparent effects on motor cognitive function, and at the same time, it has proven for over 20 years, fairly elusive in the analysis of functional connectivity. If you look at the literature, it tends to be a mess. Some people find some effects, some people find other effects. So it's a field where actually maybe cognitive architectures can make an impact. If we find something where cognitive architectures can help brain scientists, Parkinson's disease is probably our best bet. And for us, it also poses two specific challenges. And these are two specific challenges that for us were very interesting because as long as we are staying in the well-defined world of task-based fMRI data, the analysis that actually cutting edge has shown, we're okay. This is a domain that we understood and we have worked for a while there. But when we move to clinical neuroscience, all of a sudden we have to work with two different things. We have to work with pure resting state data because this is the state of the art for neuroscience analysis. No task at all. Just take patients in, let them do nothing for 10 minutes where we record brain activity. And this makes sense because it's very difficult often to ask patients to do anything while they are in a scanner. The other thing is that we had these carefully crafted regions of interest on one hemisphere, like it's common to use for us. But when we work with patients, we need to actually be sure to cover both hemispheres because you never know which hemisphere is the first to go during the onset of a disease. So this is actually how we solve the first problem, the problem of resting state. If you pay attention to Catherine's explanation, you've noticed that actually having task events trigger the events that go into this dynamic system that is the basic model of connectivity that we use to simulate the time series of connectivity in this network of regions is essential. If we don't have these task events, we cannot make it happen. But of course, in resting state, they are not tasks at all, and therefore they are not task events. So we actually tested this system in the previous 6 or 7 months, where we actually created fake task events that correspond to slow, very slow fluctuations of brain activity. We created this essentially a series of very slow regressors that correspond to brain activity at different hertz, from very, very low, slow to medium slow, but definitely not at the level of frequency of this task. It's very slow changing in brain activity. Then we discretized them, and then we fed them to our regions. We also created the equivalent, but with a phase shift, so that we can capture like pretty much all of the possible changes in fluctuation in this coordinated activity in the brain in these areas. And then we tested this model, and we actually saw that the model actually can identify still these changes in activity without a task. At this point, we're ready to identify regions, and we created this set of regions that match fairly well with the common model of cognition again, but were spread over two different hemispheres. And for every single participant in our cohort, we had actually 106 participants divided between Parkinson and controls, we were able to create a single region that spanned one side of the brain and the other. So what you see here in this kind of like small clouds of dots is actually the individual locations of these regions for every one of our 106 participants. So each dot represents the point and the brain at which we are recording our simulated brain activity. And for each dot, there is a counterlateral dot on the other part of the brain, except of course like the motor region, because participants were mostly doing something with the right hand when they were doing a task-based analysis, which is not covered in this particular case. In the other case, we use only the right hand. Okay, at this point, we've solved the two scientific tests. We're ready to go for the part, and we can ask a couple of questions. And the first question we're going to answer is, which is the best way to conceptualize the common model of cognition when we want to apply it to a clinical neuroscience population? Now, this is important because in previous papers that were published on this, we went back between different tiny versions of the common model of cognition. On the left-hand side here, you see what we call the direct version. This is the straightforward translations. Every arrow in the classic diagram of the common model of cognition is translated with an arrow in this network diagram. That is easy, and this is what we tested with the human Connectome data, and it makes perfect sense. On the right hand is actually a version of the common model of cognition that we tested, but we went back and forth whether it made sense or not, because it adds an additional level of complexity. It's a modulatory version, which the basal ganglia, the PM region, the procedural memory region, doesn't really affect working memory per se, but in fact affects the connections that go from long-term memory and procedural and perception to working memory. This is a version that is slightly different than the diagram. It's still compatible with the description of the common model of cognition. it's a little bit closer to a couple of conceptualization of the function of the basal ganglia, in particular with the models by O'Reilly and Frank and a model that I published also in the past. They see the basal ganglia as a gateway to prefrontal cortex. It's basically there to decide which part of the inputs that are possible are going to be put in working memory or not. The reason why we decided to test these two in this crucial population is because since this is the source, the procedural memory is the source of the problem in Parkinson's disease, it becomes important to have the right model to decide. Okay, at this point, this issue, which might be not particularly relevant for healthy controls, becomes important, so we decided to make a crucial test. We may do two predictions. The first one is that if really the modulatory model is what we believe is close to biology, it should be better for both controls and for patients. It should be a better fit eventually. And the second prediction is that if we analyze actually this particular modulatory connection, we should find a difference. If you really believe that this is something that really could be used in the clinical neuroscience setting, it should be able and sensitive enough to distinguish between Parkinson patients and controls. And this is kind of like we see like as a litmus test. If it works, it's really a good test that cognitive architectures could be used in the clinical neuroscience testing. If it doesn't, well, okay, we didn't lose anything, but I mean, we did a good try. So this is the first analysis I'm going to show, which is the comparison. With the comparison that was a little bit more strict than the original comparison we showed. We had less participants, so we need to do an analysis that is like allows less degree of freedoms. We use log likelihood. Essentially, we calculated how well each model fits each participant and sum up all the log likelihood. Now, it's difficult to immediately see here, but the way to compare the log likelihood is this. You just sum them all up across all participants, and the values tend to be negative, of course. The least negative value for a model is the best. If you want to transform this into a standard p-value, you can just do something like calculate the difference. We're going to call the difference between the two groups, between the two models across the two groups lambda. And there is a theorem called Week's theorem that tells us that the value minus 2 lambda is actually equivalent to the chi-square distribution. So if you plot the value of the 2 lambdas for the Parkinson's disease patients and for the controls, you can see that they correspond to p-values that are essentially very close to 0. So we know that actually these two differences are absolutely significant. We have strong evidence that the modulatory model is better for both controls and patients. So it does give us an edge in trying to understand what is the best way to conceptualize this architecture, in particular the basal ganglia, when we go into a clinical neuroscience settings. Another question is, what happens to the modulatory connection? And this is the interesting result. If we look at the modulatory connection, which are the 2 red arrows that you see in the tiny brain diagram, we see that they are both negative, which is a prediction of the models. This is something we expected. But most importantly, we see that there is a significant difference, and we did this with a permutation test to make sure we're not like, you know, fooled by the fact that maybe the data were actually like normally distributed or something like this. This is actually a pretty solid, surprisingly solid result. In Parkinson patients, the modulatory connections are much more strongly negative than in healthy controls, which is consistent with what we would expect because the death of dopaminergic neurons produces what is in the classic model of Parkinson's disease an increase in what's called the brake. So the basal ganglia inhibitory connections that Terry and Chris had previously mentioned increase to a point where actually they block all the inputs of prefrontal cortex, which produces down the line these dementia-like symptoms with the progression of the disease. So all of a sudden we have a tool that is derived from these 40 years of distilled lessons from cognitive architecture that have been applied from computer science and psychology, but to developing tools essentially to simulate behavior that can be taken back to clinical neuroscience and can let us do something that previous analysis of cognitive data was not able to do, distinguish carefully between Parkinson and controls, which was actually a surprise for us, and it was a big hit with our clinical neuroscience friends. So to summarize, it is possible to apply cognitive architectures to clinical neuroscience data, to patient data, nonetheless. And actually, it works. And this was a big surprise for us, and I think like a big test, a big testimonial, actually, to the success of this approach.
So on, I guess, the strength of that conclusion, we like to think that zooming out, we've sort of learned three main things from this line of research. The first is that human neuroimaging data is actually much easier to get now, and that access can maybe break down some of the obstacles that were preventing it from being incorporated into some of the more cognitive focused models and analyses. We think that adding this information into that whole line of work can be very useful, that we can learn a lot about architectures from that neurological data, and that those lessons can then be taken back to a more clinical neuroscience area and applied more practically beyond just theoretical accounts of the brain, perhaps in sort of a diagnostic setting even. And with that, we have a lot of people to thank.
I'll pass back control to questions to see if there's some questions for Andrea and Catherine. I'll pass back control to questions to see if there's some questions for Andrea and Catherine.
Yeah, I saw a couple. One had asked, does it make sense to assume that connectivity stays the same between different tasks? I think that's with the model that you had mapped onto the brain. So asking if it's a reasonable assumption that stays the same between tasks.
It does not. I guess that the best answer is like, it does not in terms of parameter connectivity values. But if you look at the geometry of the connectivity, in the sense like the fact that certain links are present versus other links best fit, the nectar geometry, that seems to be fairly fixed. And this was actually the biggest surprise in our analysis. And that's the reason we wanted to test the common model across multiple tasks. Because we fully expected, for instance, that the common model would work well in something like working memory, but it would not work in something like social cognition or emotion. And instead, it did, which made us wonder whether the common model is just like something that is probably the most flexible architecture that you can build with these regions and is easily deployed across different tasks. Now, the effective connectivity, the value of the links between the nodes, does change across tasks. And this will be material for a different talk. What is the relationship between the values across different tasks? But that of course makes sense. The changes because different tasks pose different demands between, for instance, like the relationship between perception and working memory, perception and action, and so on.
Great. And then John had asked how the common model mapping differed from SPAWN. If you'd have any ways to contrast that.
I think that my attempts to respond would be a mischaracterization, since we have the developers of SPAWN here. I feel that I can easily see a general mapping, and in the paper that we have out on bioarchive and is now in the second round of reviews, we make the claim that SPAWN is largely compatible with the common model. But of course, like SPAWN has a level of complexity that is much bigger. This also, if I can make a 30-second note to this, one of the questions that was asked by Maike was also the connection. It was about representation, like for instance, predictive coding and so on. One of the biggest differences between our analysis and, for instance, like a model like SPAWN is that SPAWN spends a lot of time thinking about representation, what the individual neurons contribute to the representation, and how the different areas exchange information through different representations and through spiking neurons. In our models, because we have to feed them to so much data, actually everything is abstracted to a dynamical system, and all the regions are abstracted to a single point, in essence. So there is no representation. It's pure fitting of time series.
Chris, I'm going to jump in and ask you, can you talk? So you did have a connectivity diagram. And I wanted to know if what your thought of is in terms of the compatibility with what was presented by Andrea.
Sure. Yeah, I think basically the level of detail is just much higher in SPAN, pretty much exactly what Andrea said. So when I look at the CMC, I see that complete compatibility with most of, well, at least, well, I don't know about complete, but like a lot of compatibility with the assumptions that SPAN's built on. But inside SPAN, we basically have 20 different areas, and many of them are connected to many others, and it just kind of goes into a lot of additional anatomy and so on that the CMC is kind of agnostic towards. So yeah, I think Andrea's response was a good one.
There is, if I can make a meta note to Chris's note, there is one thing that in John's very first presentation, John went through the newest level of analysis. And there is one thing that has always been in the back of my brain every single time I saw it, is that newest bands are really about time, but they seem to be a little bit like going back and forth between time and space. But when you go to the brain, like time and space become a little bit like different things. Like you can have things that happen very fast in time, but across very large areas, or things that happen over very long times, but in at a very micro level. And I think that when you reach the level of analysis that Chris and Terry do, or even when you look at very high density brain data, you need to start thinking about these two dimensions of time and space very clearly.
Yeah, I'd agree with that. And I think fortunately we've focused on tasks so far where we haven't really had to deal with that tension, but I think there definitely will be places where that's true.